morning, everyone. I'm Gigi Wu Alice from Hematology Branch and HLBI at the NIH. Today, I'm presenting our analysis of deficiency of adenosine deaminase 2, or DADA2 pathogenesis, based on single-cell RNA sequencing of monocytes. I am presenting on behalf of Dr. Naoki Watanabe, a previous postdoc in our group and who now is returned to Japan. DADA2 was first reported in 2014 by researchers from Israel and the United States. It has varied clinical manifestations. DADA2 typically presents in childhood and is characterized by vasculopathy, stroke, inflammation, and immunodeficiency. DADA2 patients also present hematologic syndromes such as bone marrow failure, myelofibrosis, or pure red cell aplasia. DADA2 is a rare autosomal recessive disease caused by loss of function variants in ADA2 gene. Mutations are present throughout ADA2 gene. Most patients have heterozygous missense mutations. ADA2 produced from monocytes acts as an autocrine factor to stimulate their differentiation to macrophages. ADA2 plays a role in maintaining the balance between M1 macrophages, which is pro-inflammatory and can produce TNF-alpha, and M2 macrophages, which are anti-inflammatory. In ADA2 deficiency, extracellular adenosine is accumulated, and patients have M1 macrophage polarization, which may promote inflammation and tissue damage. Pro-inflammatory cytokines are also increased in DADA2 patients. It has been reported that skewing of macrophage subtypes may lead to vasculopathy and inflammation in DADA2 patients by formation of neutrophil extracellular traps triggered by adenosine and secretion of TNF-alpha by M1 macrophages. Another evidence of the role of TNF in DADA2 pathogenesis is that TNF inhibitors are effective in DADA2 patients. To study molecular mechanisms of DADA2, we performed expression profiling of CD14 positive monocytes from peripheral blood of 14 patients and 6 healthy donors using single-cell RNA sequencing. We also measured plasma cytokine levels using a high-sensitive method. These 14 patients include 2 sets of siblings, patients 3, 5, and 6, and patients 7 and 8. I'm going to go through the main clinical characteristics of these patients. In this cohort of patients, disease-associated variants were found over the entire ADA2 gene, and the most frequent are 453 tyrosine replaced by cysteine, presented in 4 patients, and 169 arginine replaced by glutamine, seen in 3 patients. To be noted, we defined 2 variants that have not been reported yet. Patient 13 had a homozygous duplication of a region comprising exon 7, and patient 14 had a 34 arginine replaced by tryptophan. 7 patients displayed various hematological phenotypes, including cytopenias and hypocellular bone marrow, but no anemia, and they have low immunoglobulin levels and skin lesions. 13 of these patients were treated with TNF inhibitors. We first characterized monocyte subtypes in these patients by cell surface protein expression imputed from single-cell RNA-seq. Cell clusters were classified into 3 monocyte populations: CD14 strong, CD16 negative classical monocytes, CD14 strong, CD16 positive intermediate monocytes, and CD14 positive, CD16 strong non-classical monocytes. We found that the frequency of non-classical monocytes among total was significantly higher in patients as compared to that in healthy donors, while the frequency of classical monocytes was significantly lower in patients. Absolute monocyte counts of patients were almost always in normal range. There was no clear correlation between the frequency of monocyte subtypes and mutated domains or age of patients, or disease severity. This data suggests that the non-classical monocytes might contribute to the pathogenesis of DATA2. We then compared gene expression of each monocyte subtypes between patients and healthy donors and defined many dysregulated genes. Among them, FCGR3, which is the gene encoding CD16, a marker of non-classical monocytes, was significantly upregulated in intermediate and non-classical monocytes of patients. And the expression of CD16 was lower in classical monocytes in patients. Membrane proteins including LYZ, ApoBEC3A, and TMEM176B were upregulated in all three monocyte subpopulations. Membrane protein, which is the antisense of LYZ, was downregulated in all three monocyte subtypes. The adhesion molecular SIGLEC10 and the macrophage marker CSF1R that are known to be highly expressed genes in non-classical monocytes were upregulated in all three monocyte subtypes of patients. Among the commonly downregulated genes in all three monocyte subtypes of patients, most encoded ribosomal proteins associated with either the small or the large subunits, including RPS10, 28, 29, and RPL26. These genes are frequently mutated in Dimer-Blackfan anemia. PF4 and PPBP contributed to the regulation of megakaryocytopoiesis, were downregulated in non-classical monocytes of patients. CSF3R encodes a receptor of GCSF and critical to maturation of granulocytes, was downregulated in classical monocytes of the DATA2 patients. To explore biological and clinical relevance of these differentially expressed genes, we did ingenuity pathway analysis and gene cells enrichment analysis, and we defined that the upregulated genes were enriched in immune response pathways, including interferon signaling, antigen presentation, cytotoxic T lymphocyte-mediated apoptosis of target cells in all monocyte subtypes. These results reflect the inflammatory and autoimmune state of the disease. The GSE also demonstrated upregulated genes enriched in interferon signaling, adaptive immune system, and antigen processing cross-presentation. The top three downregulated pathways in all three monocyte subtypes were eIF2 signaling, mTOR signaling, and regulation of eIF4 and P70S6 key signaling. All these three signaling pathways regulate translation in response to stress. GSE results also show that the downregulated genes in protein translation elongation, translation termination, and translation initiation complex formation. These protein synthesis pathways were downregulated, suggesting that cells reduce protein synthesis, which demands energy process and cope with the damages of this disease during stress to diverse tissues and organs. Overall, the upregulated pathways in DATA2 are related to inflammation and autoimmunity, and downregulated pathways related to cell stress by decreasing protein synthesis. Shown on the left, the commonly upregulated genes in all three monocyte subtypes of DATA2 patients included many immune response genes, such as STAT1, STAT2, and many genes involved in interferon alpha, beta, and interferon gamma signaling, such as IFITM1 and IFITM2. Among these genes, many are pro-inflammatory genes. Like STAT1, its activation promotes M1 macrophage polarization. And these genes are enriched in interferon alpha, beta, and gamma signaling pathway. In fact, M1 macrophage markers such as CD68, IL-1B, CCL5, and CXCL10 were upregulated in various monocyte subtypes in patients, as shown on the right. Pathway analysis using the functional annotation tool DAVID showed similar results to IPA analysis. Distinctively, the TNF receptor 2 non-canonical NF-kappa B pathway was upregulated only in non-classical monocytes of patients. The TNF receptor 2 non-canonical NF-kappa B pathway involved 10 differentially expressed genes, including LTB, PSMC2, PSME2, and so on. Among them, LTB, PSMC2, and TNF SF13B expression levels are shown here. NF-kappa B associated network interactions in non-classical monocytes of DATA2 patients is shown on the right. We can see that many genes are upregulated, as shown in red, and they interact with one another in non-classical monocytes in DATA2 patients. To relate biological process to dysregulated genes in monocytes, GO enrichment analysis using TOP-GO was performed. Many enriched GO terms were closely related in individual monocyte subtypes. For example here, in classical monocytes, GO terms enriched in functions such as cellular response to cytokine stimuli, innate immune responses, and cytokine-mediated signaling. Categories of signaling transduction, responses to stimuli, and immune system were overrepresented also in intermediate monocytes and in non-classical monocytes. GSE also showed high enrichment of the innate immune system pathway and cytokine signaling in DATA2 patients. These results suggest the cellular immune and inflammatory response to stimuli were upregulated in disease. We also measured plasma levels of several cytokines in DATA2 patients and found significantly higher level of interferon gamma and TNF alpha in patients, but not IL-6 or IL-1B. Overall, we proposed a potential mechanism of pathogenesis in DATA2 that due to the deficiency of adenosine deaminase, ADA2 accumulated in extracellular microenvironment, increased adenosine acting as an agonist leading to interferon gamma production. Elevated interferon gamma activates cell signaling, leading to monocyte differentiation into M1 macrophages and the release of TNF-alpha. All these collectively contributed to inflammation and tissue damage in DATA2. In summary, DATA2 patients have higher frequency of non-classical monocytes and upregulated expression of macrophage markers. Immune response pathways, including interferon alpha, interferon gamma, and are upregulated in all monocyte subtypes in DATA2 patients. TNF receptor 2 non-canonical NF-kappa B pathway was upregulated only in non-classical monocytes. DATA2 patients have increased interferon gamma and TNF alpha levels in plasma. Elevated interferon gamma activates cell signaling, leading to differentiation into M1 macrophages from monocytes and a release of TNF-alpha. Responses to stimuli were upregulated, and protein synthesis was reduced in order to cope with aberrant immune responses and damage to diverse tissues and organs in this disease. The single-cell approach provides high resolution of whole transcriptome profiling of the disease. What I presented here is only a small fraction of enormous data that generated. Hopefully, this data of 14 patients would be useful for people who are interested and can be used for guiding future studies. Thank you for your attention, and I would like to thank all my coworkers, collaborators, and patients.